You have probably noticed that after a stressful day, your sleep falls apart. You toss around, you wake up at 3 a.m., and you cannot seem to stay down. But after a genuinely happy day, you sleep through the night like nothing can touch you. Scientists at Tsinghua University just published a paper in Science showing that this is not random. Your brain is doing it on purpose. For decades, we've known that sleep is critical for memory consolidation. Your brain replays daytime experiences, strengthening what matters and letting go of what doesn't. But what if those reactivated memories aren't just passive passengers riding through your sleep? What if they're the ones driving the car, actively deciding whether you stay asleep or jolt awake? The researchers gave mice a fear conditioning task, a mildly negative experience, and watched what happened during sleep. The result was dramatic. The animals' NREM sleep episodes, the deep restorative phase, became significantly shorter, and the mice kept snapping awake throughout the night. This is essentially the mouse version of what happens after your own terrible day. But the team wanted to see this at the cellular level. Using 2-photon calcium imaging combined with EEG and EMG recordings, they zoomed in on the basolateral amygdala, or BLA, an almond-shaped structure deep in the brain central to emotional processing. They tracked individual memory responsive cells at single-cell resolution and found that after a fearful experience, the specific cellular ensembles encoding that fear memory would spontaneously fire during NREM sleep. And each time they did, an awakening was very likely to follow. The team proved causality in 2 striking ways. First, they used optogenetics, a technique where specific brain cells are genetically modified to be sensitive to light, allowing scientists to use flashes of light like a remote control to turn those cells on, to directly activate the negative memory cells in the hippocampus or amygdala during sleep, which instantly induced an awakening on command. Second, they used chemogenetics, a similar method where targeted brain cells are modified so they can be turned on or off by administering a specific, otherwise harmless chemical to specifically silence those exact stress-related memory cells. When they did this, the sleep fragmentation completely disappeared and normal sleep continuity was restored. This confirmed that the disruption was driven by the reactivation of specific memories. And here is the part that feels like science fiction. The team built an AI-based calcium signal decoder that watched these firing patterns in real time. It got so good at reading the activity of negative memory cells that it could predict an awakening before it happened. So if negative memories sabotage sleep, can positive memories do the opposite? The answer is a resounding yes. When the researchers activated memory engrams in the BLA associated with a rewarding social experience, essentially replaying a happy memory during sleep, the animals showed significantly enhanced NREM sleep continuity, with far fewer awakenings and much longer uninterrupted sleep. But the stunning part, when the team introduced random disruptive background noise, the mice with activated positive memories slept soundly through the disturbance, as if those happy memories had built an invisible wall around their sleep. The team traced the neural circuitry behind this and found that positive and negative memory engram cells in the BLA connect to completely different downstream brain circuits. Positive engrams preferentially talk to NREM sleep-promoting regions, while negative engrams are wired into arousal-promoting pathways. It's like your brain has 2 separate telephone lines. One calls the sleep department, the other calls the wake-up department, and which line gets dialed depends entirely on the emotional color of the memory being replayed. So who decides which memories get replayed? The team mapped the chain of command and found that the hippocampus sits upstream from the amygdala and acts as the prerequisite coordinator. It is the hippocampus that determines which memories are reactivated in the amygdala during sleep, making it the true gatekeeper of whether a memory will protect or sabotage your rest. This matters far beyond the lab. When the team studied mice subjected to chronic immobilization stress, a model for depression-like states, they found that the spontaneous reactivation of stress-related memory engrams during sleep was the direct cause of severe sleep fragmentation. The implication is profound. The sleep disruptions that plague people with chronic stress, anxiety, and depression may not be a side effect at all. They may be a direct, mechanistic consequence of negative memories replaying night after night in the amygdala, like a broken record that keeps waking them up. This study reveals sleep as an active, adaptive state where the brain calculates whether to rest deeply or remain alert based on the emotional weight of daily experiences. And it raises ethical questions. How long until we can do the same for humans with stress-induced insomnia? And if such tools are developed, who decides which memories are preserved and which are suppressed during sleep? If you found this insightful, please like and subscribe to support the channel. Thanks for watching.